Good afternoon, ladies and gentlemen. Today we will start this first session of the afternoon talking about therapies. My name is Guillem Pintos. I am a pediatrician specialized in inborn metabolic diseases, working at the reference unit of hereditary metabolic disorders in Barcelona. And the session today will be the first of 3 other sessions dedicated to therapies. And we will have 4 eminent presenters, speakers. We will have Professor Eto, we will have Dr. Avasolo, Professor Giuliani, and Professor Paul Harmatz. So we will start today with Professor Eto. Professor Eto has extraordinary experience in lysosomal disease. I like to remember that Professor Eto is the director of the Advanced Clinical Research Center in Southern Tohoku Institute of Neuroscience in Tokyo, and also professor at the GK University School of Medicine in Tokyo. Professor Eto has been the president of the Japan Pediatric Society and Japan Society for Inherited Metabolic Disorders, and was the president also of the 10th International Congress of Inborn Errors of Metabolism in 2006 in Tokyo. Today, Professor Eto will talk about MPS in Japan, current treatment for central nervous system. Professor Eto.
Okay, thank you very much. Okay, thank you very much, Professor Pinto. The nice introduction. And also I would like to express a thank you for inviting me here, Professor Pinada, and also Professor Bosch and other organized members in this meeting. So I will talk about MPS in Japan and the current treatment for CNS. And this is COI. And today's topics, I would like to talk about current status MPS in Japan, distribution, the number, and mutation, Japanese MPS 1, 2, and the current status of ERT in Japan. And also we'd like to talk about CNS treatment. Number 1 is the hematopoietic stem cell therapy, intracerebral ERT, and BBB penetrating enzyme, and gene therapy. And finally, we just talk about a little bit newborn screening. This slide shows the prevalence of patients with lysosomal storage disease in Japan, supported by the Japanese government ministry. And I ran this project since 20 years. And most common lysosomal disease is a Fabry disease, second common MPS 2. And this slide shows the frequency of MPS in Japanese. Overall frequency of MPS 1 to 60,000. This is a little bit less incidence compared to those of Caucasian, but the MPS 2 is the most common among these. It's a frequency about 1 to 100,000, and MPS 6 is a very rare, 1 to 3 million. So the distribution of this pattern is different from those of Caucasian. This slide shows the distribution each type of MPS in Japanese. As more than 60% is MPS 2, and the second common MPS 3, and MPS 1 is the third common, 13%. And this distribution is quite rather different from Brazilian, Dr. Giuliani's place. They have a much higher incidence of MPS 6 and the Japanese distribution, MPS 6 is very less. Most common one, MPS 2, as compared to those of the MPS 2 is very much higher compared to those of the Caucasian and also much higher than the European people. And so this is a gene mutation MPS 1. And the common mutation Caucasian is the W402X or Q70X. But this mutation is not in Japanese. In Japanese, common mutation 744 insertion 5 or R89Q are rather common. And so the pattern of the gene mutation MPS 1. And this slide also shows that MPS 2, about 70% of Japanese patients are severe types, and attenuate type and 30%. And we have a private mutation, novel mutation in the MPS 2 and R468Q are rather common, but this mutation also different from those of Caucasian. And this slide shows the natural history of Japanese MPS 2. This reported by the Okuyama's group. And this is chronological age, and this vertical line, the vertical line also shows the developmental age. As shown here, this is the normal developmental pattern, and the missense mutation, the dotted line, null mutation type is a solid line. As shown here, at the age of 3 to 4 years, stop the intellectual development. After 4 to 5 years, start the deterioration, as shown in this slide. Patient with null mutation more deteriorated than the missense mutation. Residual enzyme activity in the missense mutation are usually much higher than those of the null mutation. So the residual enzyme activity influences the brain function as well. And this slide shows the clinical future MPS in Japanese. More Mongolian spot in Oriental MPS patient or MPS 2, most common in Japanese and Asian people. And MPS 4 or 6 is less common in Japanese and common in Brazil or Portuguese, and rather less severe form of MPS 1 and 2 in Japanese Oriental patient, maybe due to different pattern of gene mutation. So now we switch to the treatment. The symptomatic therapy, of course, important. And as a specific therapy, we discussed about first hematopoietic stem cell and the ERT current status and novel treatment, new treatment approved, and also gene therapy. So the first enzyme therapy this carried out by Roscoe Brady, as you know. And when I meet at NIH 2073, he purifies the enzyme from placenta at NIH. At that time, enzyme does not so much work because of that is a natural enzyme, the acylic acid terminated enzyme. After that, Roscoe carried out the mannose terminated glucocerebrosidase enzyme production, and also the Elizabeth Neufeld and Bill Sly discovered the mannose 6-phosphate receptor mediated system, and now the enzyme can be taken up by the mannose 6-phosphate receptor mediated so-called target therapy in the various lysosomal disorders. This slide shows the current status of Japanese patient treated by enzyme. Right now, 1,500 patients treated by enzyme. Most common one, the Fabry disease, almost 1,000, Gaucher 100, MPS around 200, and Pompe 100, and LAR deficiency, currently MPS 7 treated by enzyme. And this is first enzyme therapy, MPS 1. First, this enzyme developed by Emil Kakis. He'll also give a talk today. But Emil, at that time, I visited his lab, 1998, almost 23 years ago. He purifies enzyme in such a small lab in the UCLA Harbor Hospital. At that time, I visited him, and he carried out the first clinical trial of MPS I alderazime, and he's the first man to treated by enzyme MPS I enzyme. And this is the further. He's actually the president of U.S. MPS Society, and this is Emil. We are young at that time enough. After that, 20 years later, I met him. He just graduated from college. He looks very good, as shown here. And I met him 2017 in Tokyo. He's okay now. But unfortunately, I treated first patient, MPS I, Hara-Shire syndrome, and she's rather severe at home. And but I treated her. So Emil developed the enzyme, and she went to the biomarine, and he developed the aldorazime enzyme. So this is a biomarine enzyme also. We treated MPS 6 cases, MPS 6 marotolamine, and this patient treated 1 month of age. And her brother, elder brother, started at the age of 5. Look at their face, and she looks a normal face when treated for 3 years by enzyme. And also she can raise her hand, but her brother does not so much well. And also this is a developmental height development, and elder brother does not gain the height after 4 or 5 years, and deteriorated rather slow in the development. And his sister growth development better after ERT, but becomes slow after 5 years old. And this is why because the bone deformity already appeared after, although she's treated more than 3 years, but as shown here, still bone deformity already present, and also vertebrae is also deformed. So the ART does not work so well on the bone. This is the first patient treated by enzyme. Treated enzyme for 13 years, died 4 years ago, and unfortunately she died. And we carried out autopsy, but the enzyme therapy treats the skin becomes soft, and softening joint tendon, decrease cardiomegaly, hepatosplenomegaly, or increase appetite and QOL. But the autopsy finding in the brain showed that massive accumulation of MPS also subunit mitochondrial ATP synthase, and unfortunately she died. So, as shown, and therefore the efficacy and limitation enzyme therapy, no improvement of cardiac valve disease, joint range motion, and skeletal disease and CNS. So ERT can prevent the onset of the disease future, thereby dramatically altering natural history of the disease. Actually, different phenotype by enzyme therapy. So CNS is a target for the treatment. That's important issues. And the recently number of the treatment procedure has been introduced. The hematopoietic stem cell therapy has introduced almost 40 years ago by the we'll tell later. And enzyme therapy, actually 2 enzyme protocol, intrathecal or interventricular injection enzyme already carried out MPS 1 and 2, and 3A and NCL2 MLD, and also BBB penetrating enzyme, that is transferrin receptor mediated system. And then recently gene therapy, we already discussed about this kind of the important issues. And after my lectures also, there are several important lectures will be prepared. So direct injection into the ventricle by airway gene therapy, or lentivirus gene therapy, or the intravenous injection, AV9 gene therapy. So this slide shows that the number of the cases hematopoietic stem cell transplantation in Japan. Professor Kato described 2013, 100 cases treated by bone marrow transplant and 57 cases in the MPS II and MPS I, 24 cases. So more than 50 cases, 50% is MPS II. And this is the slide shows the hematopoietic stem cell in MPS I and in Japanese patient treated by the bone marrow transplant. If you treated before 2 years old and mental development catch up to the normal pace, and after 2 years, also that's difficult to catch up. So Professor Hobbs and Professor Whitley and Kribit carried out this kind of important trial for the bone marrow transplant and various lysosomal disease. Dr. Tanaka, unfortunately, she died several years ago, but she reported long-term efficacy of hematopoietic stem cell transplantation in MPS II patient. She collected 21 cases, and she concluded that ADL maintained after around the baseline level, and cryophomic changes and ventricular dilatation on brain MRI was improved in 1917, 4 to 17 patients respectively, and the stabilization of brain atrophy. Also, cardiac valve regurgitation was diminished, 20 to 63 cases, and urinary gag also decreased. And in the questionnaire analysis, speech deterioration observed 12 out of 19 untreated cases, 1 to 7 patients in treated cases. So hematopoietic stem cell transplantation show effectiveness toward brain or heart involvement when performed before sign of brain atrophy or valvular regurgitation. We considered bone marrow transplant is worthwhile in early stage of the disease for patients with MPS, even MPS II, particularly CNS symptom if treated before 2 years of old. This is the same situation in MPS II. 1. So now we must two type of the CNS treatment was approved in Japan this year. That is intraventricular ERT once a month, injection, intraventricular injection, or Laplace weekly injection. And also BB penetrating enzyme, anti-transferrin receptor antibody binding enzyme, so-called ISCARGO, approved, developed by the Japan Chemical Research. Also the Professor Giuliani will also talk about more detail later. So this, before talking about the clinical trial of ventricular injection of the enzyme MPS II, we carried out almost 10 years ago, ERT, intraventricular ERT MPS II in the MPS II mice. And we injected the IDS enzyme 20 microgram every week, 4 times, and the different region also increased the huge amount of the enzyme activity in different region. Also, the intraventricular injection of the enzyme also increased amount of the IDS enzyme activity and different organs, eye, lung, heart, liver, kidneys, spleen, testes. All the organ also increased amount of the enzyme activities. So this is because the enzyme transfer from the cerebrospinal fluid to venous blood via arachnoid granulation, and then increase IDS activity in the different regions. And also this slide shows also the improved mice face, and also the enzyme distributed in different regions of the brain as well. So another important papers, the Dr. Okuyama's papers, and they also inject the intracranial hydrosulfatase beta enzyme in the MPS mice model, and they demonstrate that heparan sulfate in CSF is can be used as a clinical biomarker for the evaluation. So one shot of the enzyme, the intraventricular enzyme, also the increased amount of the intraventricular CSF heparan sulfate also parallel to the brain heparan sulfate. So heparan sulfate can be used as a biomarker for the efficacy or evaluation of efficacy of the enzyme treatment. So this slide shows the change in heparan sulfate in CSF patient with MPS II after interventricular ERT. As shown here, the follow-up the 100 weeks, and then heparan sulfate CSF content is also declined and stabilized. So also this slide shows the developmental age after the treatment, intraventricular enzyme therapy in the 6 cases. So most of the patients stabilized in the developmental age measured by the Kyoto scale score. Another important technology is BB penetrating enzyme therapy using anti-transferrin receptor antibody binding enzyme, that is receptor-mediated transcytosis. There are a number of the possible ways to treat to cross the blood-brain barrier. These are transferrin receptor antibody enzyme, binding enzyme, melanotransferrin receptor, or apolipoprotein E, or LDL receptor related protein, or receptor for advanced glycosylation end product, or insulin receptor system, or Fc gamma receptor system. So among them, the transferrin receptor antibody binding enzyme has seemed to be most successful right now. So J-Brain cargo developed by the Japan Chemical Research Company, and they made the enzyme human IDS bind to the anti-human transferrin receptor antibody, and this enzyme can cross the blood-brain barrier. And this slide shows a clinical result, so-called new enzyme JR141. This slide shows this slide shows a severe type and attenuated type, and measures the CSF heparan sulfate. And as shown here, after treatment by enzyme, the heparan sulfate, CSF heparan sulfate decline, and also in attenuated subject also decline in the CSF heparan sulfate. And 4,000 gram per liter heparan sulfate CSF is a critical amount to distinguish severe or attenuated form of MPS. And this is another result, and treated by new enzyme, and CSF heparan sulfate go down, and then switch to the usual enzyme and go up again, and then switch to the new enzyme or go down the heparan sulfate CSF. So that means that this enzyme also acts to the brain efficacy. So this slide showed also the important. This is a clinical result about 28 cases, and check the development curve by beta scale development assessment. So green cases show the improved, blue one stabilized, and red one deteriorated. Many of the patients treated by the new enzyme JR141 show the improved or stabilized. And also this is also important, two siblings, that one elder one is treated by usual enzyme and follow up the development. And the new enzyme treated one shown the green line and chases the normal development. Again, this enzyme very much effective to the brain. And so this new enzyme also not so much serious adverse event. So this event can be manageable. Now finally, now we'll talk about a little bit gene therapy. We'll discuss later more detail as shown here. So now it's two types of gene therapy is available: lentivirus AAV gene therapy, and detail will be presented later. Yesterday also presented very much precisely. And we carried out almost 6 years ago hematopoietic stem cell gene therapy, correct the neuropathic phenotype, murine model of Mps Ii, using the lentivirus vectors. We demonstrated that the, we sorry, we demonstrate that lenti, sorry, we demonstrate that lentivirus hematopoietic stem cell gene therapy corrects neuronal manifestation by ameliorating lysosomal storage autophagic dysfunction in the brain MPS II mice, and IDS transduced hematopoietic stem cell increase enzyme activity both in various organ and also CNS. Now we are going to do the clinical trial using lentivirus vector in Japan. And so now is yeah, lentivirus gene therapy, every gene therapy now is carried out in different companies, and also editing gene therapy also described detailed yesterday. And so this slide shows that now it's lysosomal disease can be treated number of the possible way, and the bone marrow transplant has been carried out first, and then second the ERT, and then chemical chaperone, but not in the MPS, but the substrate reduction also available in the different type of the lysosomal disease, and gene therapy are now very much explosive right now. And however, the early treatment is very important. So even though that they are very much effective, but in Japan, we started the newborn screening lysosomal disease and SMA and immunodeficiency, ADR, start from the south about 10 years ago, now expanded to different regions. And right now we carried out personally, we carried out 7 lysosomal disease by tandem mass screening. We carried out also SMA newborn screening. So this is very much important to do this kind of project as well. And any other country also started this kind of the newborn screening. Okay, thank you very much for attention. And if you have any questions, I'm very much pleased to accept the question. Thank you very much.
Thank you very much, Professor Reto, for this very interesting review and comprehensive review. I would like to ask you, what do you think with respect to MPS II? If we have a very early diagnostic, the hematopoietic stem cell transplantation for you will be first line treatment?
Oh, yes. Actually, right now, as shown in the lecture, that we carried out the almost 50 cases treated by bone marrow transplant. But right now, bone marrow transplant have some effectiveness for the treatment. But that's some risk by the bone marrow transplant. So right now, the new enzyme therapy, 2 types of the new enzyme therapy, as I discussed already, as shown here, we already presented the 2 types of the CNS therapy. Maybe we treated by the new enzyme therapy now. So before the new enzyme, usually we do with the usual enzyme elaborate treatment. So that right now is a new protocol for the NPS II. First, we treated the transferrin receptor mediated or intraventricular enzyme therapy right now. So we started now since 1 year ago this kind of the new enzyme protocol procedures.
Okay, that's very interesting, very interesting. Thank you very much. So, okay,
thank you very much.
Yeah, thank you. Now we will go to our second speaker, that is Dr. Ivani Avasolo. Dr. Ivani Avasolo is the head of drug delivery and targeting group in the unit of nanomedicine in Vall-d'Hebron Research Institute in Vall-d'Hebron campus, Barcelona. Dr. Avasolo is a senior researcher with a long trajectory of developing novel nanotechnology-based treatments for cancer and lysosomal storage disorders. She is currently leading several projects, mainly based in the use of extracellular vesicles for treating Fabry disease, Sanfilippo A syndrome, and also pycnodysostoses. The research career of Dr. Avasolo is supported by her publications in the field of cancer and lysosomal storage disease, and she is also co-inventor for different patents, 3 related to therapeutic products and methods in lysosomal storage disorders. Today, the presentation of Dr. Avasolo will be the extracellular vesicles for improving the ERT. So the floor is yours, Dr. Avasolo.
Thank you very much. Can you hear me?
Yes, very well.
Okay, perfect. So thank you very much for the opportunity that the MPS Society has given me to present our results to all of you. So as Professor Guillem Pinto said, my name is Ivani Avasolo. I'm the leader of the Drug Delivery and Targeting Group within the Vall-d'Hebron Institute of Research in Barcelona. This center gathers together most of the clinical and also preclinical investigation that is conducted at the Vall-d'Hebron Hospital. You may already know that the Vall-d'Hebron Hospital is the main hospital complex in Catalonia. It's like the flagship of the public Catalan health system, and it has more than 1,000 beds, 7,000 professionals working there, and it's a reference center for many rare diseases, including MPS. So what I believe is really important also in this hospital is that it's one of the main top hospitals in Spain in terms of the number of open clinical trials, and many of them are actually in phase 1 and phase 2 early phases of the clinical development. So in this environment and this scenario, the group that I'm leading, the group on drug delivery and targeting, we try to implement novel treatment systems based on nanotechnology, and we do that by studying highly prevalent diseases, oncology mainly, but also rare diseases and specifically lysosomal storage disorder. And this is why I'm going to start my my presentation. I think I need to have the control. Okay, here. Okay. Let's go back. So this is why I'm going to start my presentation by giving you the motivation or why we believe that the use of nanotechnology and nanomedicine can actually help in the development of new treatments in lysosomal storage disorders, and specifically for the enzyme replacement therapy. And we have a trajectory of several years working with different synthetic and nanoparticles, liposomes or polyelectrolytic complexes, even pegylation of the enzymes. But today I'm going to focus my presentation on the use of natural nanoparticle, extracellular nanoparticles derived from living cells. And we have been developing different type of assays using GLA-A, alpha-galactosidase A containing extracellular vesicles for the treatment of Fabry disease. And I will have also one single slide on the results that we are having with the SH containing extracellular vesicles for the treatment of Sanfilippo A syndrome. So as I said, this is going to be a very short presentation. I won't have much time to go into the explanation of which are the different drug delivery systems that can be used for the improvement of the enzyme replacement therapy, but we have a very recent review that you can go and check how we are comparing what has been published for the Fabry disease treatment we use in nanotechnology-based therapies. And also all the results that I'm going to show you in here have been already published by our group in this recent publication in the Journal of Extracellular Vesicles. And as I tend to speak very fast, and I do really want you to have a clear view of what we are having in the lab, and it's very technical also. I'm numbering all the slides, sorry, here in the top corner. So if you have any questions, or you have any concerns, or you want to have more explanation in certain slides, just keep this number in mind, and then later on the chairman of the session, Professor Pintos, will drive us the questions you may have. So just to start with, nanomedicine in the field of drug delivery systems is based on I believe that the presentation that I'm showing you here is not the one that I sent the last time to the organizers, but however I will take it from here. So the nanomedicine, as I said, in the field of drug delivery systems is based on the use of nano-sized carriers that is delivering an active cargo. And the main issue here is that taking into account that the cargo is really small and the carrier is much bigger, and they have different biological properties and entities, the important thing is that having both of them together, you have a much bigger complex between 20 to 200 nanometers inside. And this difference in the size of the cargo versus the nanoparticle and the composition gives many advantages to the whole complex. And this is what nanotechnology is taking advantage for improving the solubility, the stability, biodistribution, etc., of different systems. So you know already that the nanotechnology has been used to treat different diseases, including also the use of the vaccination, that you know that some of the vaccines that are available for the SARS-CoV-2 are based on the use of nanoparticles. So the same strategy was meant to be used in the enzyme replacement therapy. Why? So this is the slide that is summarizing this. In the normal situation, you know that the enzymes are produced in the plasmatic reticulum. They go to the Golgi apparatus, and there they are glycosylated. They can then move forward thanks to the expression of the internal mannose phosphate receptor to the lysosome, and there they can have their active role being performed. But you know that up to 30% of the enzymes that are being expressed and synthesized in the plasmatic reticulum, they go straight to the extracellular space, and from there they can be uptaken by this cell line or a neighboring cell through the bind and the internalizations to the mannose phosphate receptor. So this is the natural situation, and this is what the enzyme replacement therapy relays on. So we put the recombinant enzyme in the bloodstream, and this enzyme, if it's properly glycosylated and we have the presence of the active mannose phosphate receptors in the surface of the cells, then the therapy is active and is working. But you need to take into account that you need both things. You need to have a proper glycosylation of the enzyme, and you also need to have the expression of the mannose phosphate receptor on the cells. So instead of this, when we are using a nanocarrier to deliver this recombinant enzyme, what we can do is we can protect the enzyme from the degradation from serum proteases, for instance, and we can also protect them from the generation of autoantibodies. Both things, the presence of the proteases and the presence of the neutralizing antibodies, are usually reducing the efficacy of the system of the recombinant enzyme, because they tend to stay shorter times in the blood circulation. But if we deliver this enzyme with the protected, we can increase therefore the life in the bloodstream and also the efficacy of the system. But not just that, we can also get rid of the need of this proper glycosylation for the internalization, because the nanoparticle is going to enter from a mannose phosphate receptor independent way, and this way we can also improve the biodistribution of the system, because regularly the enzymes, the naked enzymes, will go mainly to the tissues with a high expression of the mannose-phosphate receptor. But in this case, when we are delivering them in a protected shield, this internalization is avoided, so we can also get more enzyme in other tissues with lower expression of the mannose-phosphate receptor. And not just that, but we can also, if we add the proper targeting moieties on the surface of the nanoparticles, for instance, these anti-transferrin antibodies that Dr. Eto was explaining earlier, we can drive these nanoparticles towards the blood-brain barrier, make them able to cross this barrier, and then release the recombinant enzyme in the brain parenchyma. So as I told you before, we've been working with different synthetic nanoparticles. In the review that I mentioned previously, you can find a revision of them. But today, I'm going to introduce you what we have been doing with the natural nanoparticles. And natural nanoparticles, they have a generic name, is extracellular vesicles. But within this generic name, we have different types of particles. Those are all secreted by cells. We can have apoptotic bodies that are generated through the apoptotic fragmentation and bleeding of the cells. They are usually very big in size, from 1 micron to 15 microns. And then we have microvesicles and exosomes. Microvesicles are this one in green in here. They are generated by the fusion and outward burden of the plasma membrane to the extracellular environment, and they have between 150 and 1000 nanometers in size. And exosomes are much smaller. They have between 30 and 100 nanometers, and they are secreted through the fusion of the multivesicular bodies with the cell membrane. So they have somehow a common biogenesis with the endosomal pathway and the lysosomal pathway. This is probably why we can we have an increased amount of lysosomal proteins within these exosomes, as I will explain to you later on. So both system, microvesicles and exosomes, can be taken by recipient cells. So it's a way of cells of transferring some content from one cell to the other, or to be able to communicate with them. So it's a natural system that can be also used as a drug delivery system. And this is the system that we were using. And here you have a summary of the advantages of using extracellular vesicles. They are biocompatible, biodegradable. They are not toxic. They are non-immunogenic. They are stable in biological fluids, and they can be easily generated because most of cells, especially cell cultures, are producing extracellular vesicles, and we can isolate them from there. So that there have been described different methods to load a cargo into these extracellular vesicles. The most classical way is to first isolate the extracellular vesicles and then load them mechanically or by electroporation with the cargo or the molecule that you want to put into them. These are the top-down methods. But we can also, as we are using cells for obtaining extracellular vesicles, we can also modify genetically those cells and make them produce extracellular vesicles that already contain inside the cargo that we need. And this is the approach that we followed. We used an expression vector containing the alpha-galactosidase A GLA gene coded, plus an histidine tag that we later used to purify the soluble protein. So cells were transfected, and in the conditioned media of these cells, we could find the soluble protein, but also extracellular vesicles that contained the GLA enzyme, as I will show you later. Sorry. So just to make you aware that we were not the only one working in extracellular vesicles, there are other lysosomal storage disorders that have been explored also for this kind of treatment. And we have the work by the group of Natasha Batrakova and Chaper Hill that worked in Batten disease, and also several publications are going on also in cystinosis, Gaucher disease, and very I think earlier this year, also in more disease. But in our system, as I told you, we were working with CHO cells, stably expressing the GLA with this MYC histidine tag. So we had the cells and we collected the media of these cells. In the media, we had both things, the soluble protein and the extracellular vesicles. The soluble protein was purified by affinity purification, and the extracellular vesicles, in this case, by neutralization-based precipitation methods initially. So what we are obtaining here were, as you can see, both things, microvesicles with bigger size and also exosomes. We did some nanoparticle tracking analysis, so we could find that most of the nanoparticles were actually exosomes, and we also found that they contain indeed the markers of the exosomes, like the CD81 and TCH101 in here, and they did not contain that much the cytoplasmatic proteins such the beta tubulin. We also checked for the expression of the GLA. As expected, it was really high in the extracellular vesicles. We called them AVGLA, but we also found, interestingly, some other lysosomal proteins such the cystinosin or the GBA. So this was very interesting also for us to find this out. And the important thing here was that we also checked for the activity of this enzyme that was being delivered in these extracellular vesicles. And if you can see, if you see here in the graph, we had the GLA in the extracellular vesicles, and the activity was really high, much higher than the activity in the soluble GLA, and also much higher than the activity of agalsidase alpha, which is the recombinant enzyme that is being used in the treatment of Fabry patients. And also the activity was considerably high, or was absent in the extracellular vesicles of cells that haven't been transfected with the GLA coding plasmid. So you could say, okay, this could be because along with the extracellular vesicles, you are precipitating also the soluble fraction and the soluble protein, but this was not the case. We demonstrated that by using an alternative method of purification of the extracellular vesicles. So by ultracentrifugation, we could find in the first step that we had actually both things. We had the extracellular vesicles and the soluble protein, but the activity was mainly in the found in the fraction that was purified after the second ultracentrifugation with just the GLA that was bound to the membrane, as you can see in here. So far we found that we could purify the extracellular vesicles, that the GLA was inside these extracellular vesicles, and that the activity of the enzyme was pretty high in these extracellular vesicles. So then we checked whether they were able to actually deliver this enzyme to other cells, and we did that by doing internalization assays in different cell lines. We have here cells from the kidney, because you know that one of the main affected organs in Fabry patients is the kidney. We want to check that, and we saw that there were extracellular vesicles in these cultures, and we also checked that in primary cultures of endothelial cells. And both of them, we could see that the internalization process was occurring. And we also, by STOM images, we tracked individual vesicles, and we found that they were actually colocalizing in the lysosomes with the LAMP1 marker. So they are reaching the lysosome. We also checked with whether the internalization process was mediated by endocytosis, and for that we used different inhibitors of the endocytic process, also inhibitors of the macropinocytosis, and we found that actually compared to the system that to the test tube that was not using an inhibitor here, when these endocytosis inhibitors were being used, the internalization process was being reduced. Also, we did the assay of mannose 6-phosphate competition for the soluble protein, as expected when we were using the mannose 6-phosphate competition, and the receptor was not available for the soluble protein. We couldn't find any enzymatic activity in the cells compared to the control. But when we were doing so with extracellular vesicles containing the GLA, we still had a lot of activity, meaning that we were getting rid of this internalization through the mannose phosphorase receptor. So this was also very, very, very good in this case. But we also wanted to know whether this was efficacious, whether these extracellular vesicles were able to reduce the substrate accumulation in the lysosomes, and the substrate that usually accumulated inside the lysosomes of these Fabry patients and Fabry disease models is the GB3. So we conducted this experiment. First, we took primary cultures of Fabry knockout mice. So it means that these cells didn't have any GLA expression. We incubated them with GB3, which is the substrate that was fluorescently labeled. Once the cells accumulated this GB3 inside the lysosomes, then we performed the treatment with different GLA or extracellular vesicles with the GLA. And these were the results. So this is an incubation curve. So we were trying to see whether we were reaching 100% of loss of the GB3 signal in the cells. And you can see that for really low concentrations of the GLA, the extracellular vesicles were doing much better in reducing the GB3 deposits than the free enzymes, either the one that we were purifying or the one that we were getting from the clinics. So good results also of efficacy in vitro, and then we moved for the in vivo assay. So what we tested first was the biodistribution of the system. We labeled the extracellular vesicles with a fluorescent marker, administered them intravenously through the tail injection in mice, and we could see that, as expected for a nanoparticle with a size of 100 nanometers, many of the extracellular vesicles were getting into the liver and the spleen, but other organs such as the lungs or the kidneys were also getting some of the extracellular vesicles that were being administered. And we also checked that this fluorescence was going along with the protein, and we could see by immunohistochemistry that we had high levels of GLA in the liver. And regarding the enzymatic activity in this liver, where this has this huge amount of the GLA, we could also see that there was a high content of enzymatic activity, much higher than in the animals treated with a free enzyme, and the same situation also in the kidney. And this is important because in the kidney, we were able to reach the levels of enzymatic activity as in the wild type mice. So with this, we'll move into efficacy assays after repeated administration of the extracellular vesicles or the control with the free drug. And we did the analysis of the GB3 levels. The components that we were administered the extracellular vesicle had higher activities, and we could also see that we had higher activities in the kidneys, as you can see here, because we were delivering actually more protein. And also in the plasma, we could see that the extracellular vesicles were able to reduce the GB3 levels to the levels seen in the wild type animals. This was not the case for the free enzyme. And in the kidneys, which is the target organ, as I told you, for the Fabry disease, we were with extracellular vesicles be much more effective than the free enzyme. So then we were asking ourselves whether the system was going to arrive also to the brain, and for that we conducted this experiment where we had positive control, meaning that we were really opening the blood-brain barrier by an intraarterial administration, and here also by fluorescence we could see that there was accumulation of the fluorescence in the brain. Not in the case of the intravenous administration of the GLA intravenously here by ex vivo whole organ imaging, but then when we moved into the confocal images, we could really see that in both cases with the intraarterial administration, where we were really broken the blood-brain barrier, we could see red signal corresponding to the extracellular vesicles in the brain parenchyma, but we also found some scarce red signaling in the brain parenchymas of the animals that were receiving intravenous administration of the extracellular vesicles. enzyme that was not doing any change in the GB3 levels of the animals. When we were administering the GLA into the extracellular vesicles, we could find a reduction of up to 30% of the GB3 levels compared to the wild type. So these were really good news, and we wanted to see if we could extrapolate this to other diseases, and this is what we have done so far. We were able to also isolate extracellular vesicles with the CSHSH enzyme defective in the Sanfilippo syndrome. So we had the enzyme there, those extracellular vesicles were internalizing, and we can also find that the activity of the enzyme is much higher in this situation. We haven't done in vivo assays yet. We are willing to do that, but we expect that in the next month we will be in a situation where we could, complete, or at least start with those experiments. So just as a summary, because I know that I'm running out of time, I believe that I could explain, or at least try to convince you that extracellular vesicles are a good vehicle for lysosomal enzymes, because they increase the activity of the enzymes, and this is something that I haven't got time to explain to you, but they can also be lyophilized, so you can have those as an out-of-the-shelf treatment to give to the patient. So it also facilitates the future translation of this into the clinics. In Fabry disease, we have seen that we can improve the biodistribution, the efficacy, and in Sanfilippo A, we are still trying to go ahead with the in vitro and in vivo assays. So this is it. This is all from my side. I want to end by acknowledging all the participants in the work, especially the Kim Seras did all the isolation of the extracellular vesicles and the characterization of this system, and here Vanessa and Mark worked a lot in the in vivo and in vitro systems and testings of the extracellular vesicles. And I think that this is all. If you have any questions, I will be happy to take them through the chairman.
Thank you very much, Dr. Avasolo. I think that are very exciting results. So while I wait if there is any question from the audience, I would like to ask you, you said that certain types, different advantages of using these natural vesicles compared to the artificial nanoparticles. Do you think also that this is like that in terms of directing the target of your organs or treatment? These vesicles are also advantage in this directing treatment?
Yes, so one definitely this is something that can be done. So you can use the natural tropism of extracellular vesicles. They tend to go to the same tissues where they were originated. For instance, exosomes from the bone will try to go to the bone of the animals that are being administered, or the same for exosomes of lungs and lines, for instance. They will tend to go to the lungs. You can use the natural tropism of the extracellular vesicles, depending on the origin of the cell, but you can also modify those cells genetically so they can actually include some targeting moieties on the surface of the extracellular vesicle. Or you can do this top-down approach where you take an extracellular vesicle and you modify by adding covalently, for instance, a peptide that will drive this vesicle to a certain cell type. So yes, this modification on the surface and this targeting activity can be done by several ways.
Okay, I have here some questions from the audience. One is, do you know or can you speculate why the enzyme activity is so much higher when in the extracellular vesicles? And also, what antibodies did you use for immunostaining in the Fabry research?
Okay, so regarding the first question, we don't really have an answer to this. We know that in general, lysosomal enzymes are very unstable and very level, and that when they are combined with lipids, this stability increases. So the fact that these extracellular vesicles have a high component in lipids, and that the GLA is very close to this lipid component, we believe that is the reason why we have this increase in the activity. So we are increasing the stability of the enzyme, and this is why we see a higher activity. But we are still doing experiments and research on this line. And then regarding the immunohistochemistry of the GLA, it's true that it's a difficult. I mean, there are not many antibodies that are working well for the immunohistochemistry. I believe that the one that we were using here was bought to Sigma. It was a commercial antibody that can be. I mean, the references are already in the publication that I told you, the one by him.
There are several other questions that I will direct to you because we are now just following the time, but I will collect these interesting questions and we will try to respond during the day. Okay.
Thank you very much, Dr. Pintos.
Thank you very much. So we will now go to our next speaker of today, which is Professor Roberto Giuliani. Roberto Giuliani is professor of the Department of Genetics, Medical Genetics Service in Hospital Clínicas de Porto Alegre and House of Rares in Porto Alegre, Brazil. Professor Giuliani is a medical geneticist with a specialization in inherited metabolic diseases. He is the editor-in-chief of the Journal of Inborn Errors of Metabolism and Screening and chairman of the Latin American School of Human and Medical Genetics, coordinator of the Brazilian National Institute of Population Medical Genetics, and member of the Brazilian Academy of Sciences. The Professor Giuliani's main interests are concentrated in screening, diagnosis, and treatment of inborn errors of metabolism, particularly MPS and other lysosomal storage disorders, being author of more than 500 scientific papers. So today, the title of the presentation of Professor Giuliani will be the fusion proteins, a new generation of intravenous ERT. So Professor Giuliani, thank you for being with us, and the floor is yours.
Thank you, Gillian, for this kind introduction. Thank you to the organizer for inviting me to be in this round table with some so distinguished colleagues. It was very nice to see the perfect introduction by Professor Eto, and now this exciting presentation by Dr. Avasolo. And so I will focus now on this fusion proteins as a new generation of intravenous ERT. As Gillian just mentioned, I am Roberto Giuliani from Porto Alegre, Brazil. Here are my disclosures. So we heard from Professor Eto about the treatments, specific treatments for mucopolysaccharidosis. I will just remind about standard intravenous enzyme replacement therapy. The story started in the 90s with Gaucher disease. They initially extracted the enzyme from placenta. They need a lot of placentas to produce 1 bottle of enzyme to administer to the patient, but later new technologies of genetic engineering and cell expression systems was able to provide a large quantity of enzymes to treat several diseases. And you have treatment for MPSs since the early 2000s, so almost 20 years now on the market. So this standard intravenous enzyme replacement therapy is administered peripherically, but has some limitations. And one of the major limitations is the fact that the blood-brain barrier does not allow this enzyme to bypass it. So the enzyme does not reach the brain. And you have several MPSs that have CNS involvement and need to have enzyme into the brain. So work about this has started long ago. This presentation was from 1992 by Bill Partridge and also Ruben Boado and others about this new approach of drug delivery with molecules that can bypass the blood-brain barrier. They are also called Trojan horse because they are able to bypass the barrier and get into the CNS. The fusion protein has a dual receptor targeting ability, and the first developments were made by this company from California, Harmatz. Already in 2007 they published this paper about genetic engineering of a lysosomal enzyme fusion protein for targeted delivery across the human blood-brain barrier, this focusing on lysosomal disorders. And basically this fusion protein has two parts. One part has an antibody that connects to a receptor at the blood-brain barrier, and the other part has the enzyme that you want to be active into the CNS. So this antibody part targets, in the case of this Harmatz product, targets the human insulin receptor that is present on the blood-brain barrier, and the enzyme has the mannose 6-phosphate receptors that is able also to deliver it to somatic tissues. So what happens is that when these fusion proteins with this antibody that is recognized by the human insulin receptor arrives to the blood-brain barrier, it is recognized and then is allowed to bypass by receptor-mediated transcytosis and then can get into the CNS. You can see in this preclinical data about in rhesus monkey about the passage of through the blood-brain barrier of this product produced by Harmatz and the AGT-181 that was targeting the mucopolysaccharidosis 1. You see in the left side the normal enzyme, Aldura enzyme, when you do not see any impregnation in the brain, while using this fusion protein, the AGT-181, you see that the brain is impregnated with the enzyme, so it is able to cross the blood-brain barrier. And regarding the somatic uptake, you see either on the Aldura enzyme in the left or AGT-181 in the right, you see that is able to reach the somatic tissue. So the information that comes from this preclinical study is that the difference is that the AGT-181 can reach the CNS. So this brain-penetrating intravenous enzyme replacement therapy is administered peripherically, but has the ability to bypass the blood-brain barrier and reach the brain. So with this, after this preclinical studies, clinical study, a phase 1 trial was conducted in Brazil with this AGT-181 in MPS I patients previously naive or on ERT with laronidase. They had weekly infusions of this AGT-181 for 52 weeks, where we observed safety, pharmacodynamics, and exploratory efficacy. And this was the first patient that was dosed at our center. And what we observed very briefly was that there was a stabilization on the urinary gags. That patients previously on laronidase, they maintained the low levels of urinary gags along the study. But we observed also some improvements, further improvements, like the liver further decreased by around 11%, the spleen further decreased about 14%. The shoulder flexion further increased around 10%, and the shoulder extension also further increased about 9%. And regarding the neurocognitive results, we observed a stabilization or improvement versus the expected decline based on natural history. You see on the left part of this slide, the natural history of the cognitive decline. You see the upper, the Bailey for multiple domains, and in the bottom, you see the Bailey for decree. And you see that there is a constant decline. And what we saw observing the AGT-181 was an stabilization of these parameters along the 52 weeks. So it's quite encouraging results. And our conclusions after that trial is that the safety and tolerability of long-term AGT-181 administration was demonstrated. Drug-related events include the infusion-related reactions and transient hypoglycemia, a very small proportion of patients. The hypoglycemia related to the human insulin receptor, but very well, easily controlled. The decree is stabilized with no net change after 52 weeks on AGT-181 based on the Bailey and Kaufman and Vineland. And the cortical gray matter, I did not show these results. Cortical gray matter volume is stabilized with 52 weeks on therapy. Also, the urinary gags were stabilized. And so this treatment appears to bring further somatic improvements in subjects previously treated with hyaluronidase, based on the further reduction in liver and spleen volume and increase in shoulder flexion and extension range of motion. And interesting, the anti-drug antibody formation with AGT-181 treatment is comparable to the profiles with hyaluronidase. So it's very encouraging results. We published this back in 2018, but and the natural next step would be a phase 3 trial, but it did not take place. And the company Armagen is now part of JCR. And I will tell what JCR is doing on this area. So now we'll move gears for the MPS II. And these studies were developed by the Japanese company JCR. And basically, it's a very similar approach. There is a fusion protein. The antibody is aimed to bind the transferrin receptor at the blood-brain barrier, and the enzyme is not similar to hyaluronidase, but is similar to idursulfase. So this product, it's able to target somatic tissues with this dual targeting the transferrin receptor and the mannose 6-phosphate receptors, and can reach somatic tissues very well, but is also able to, with the transferrin receptor, to bypass the blood-brain barrier and reach the brain. So this was planned, it was performed a phase 1 trial in Japan, and I will report the data that we were able to do in Brazil, the phase 2 study with this product. Patients, they should be on idursulfase for 6 months or naive, and so they were screened and enrolled in one of the 3 treatment arms: 1 milligrams per kilo, 2 milligrams per kilo, or 4 milligrams per kilo, and received this for 26 weeks. So the primary objective was safety. Secondary and exploratory objectives were CSF, serum, and urinary heparan sulfate and dermatan sulfate, neurocognitive testing, and also adaptive behavior testing and others measures. So the patients were planned to have 6 patients from 0 to 3 years, 6 patients 4 to 7 years, and 6 patients 8 years or older. In fact, we have at the end 19 patients. And so the results, well, we can see the CSF concentrations of heparan sulfate in patients you see in the 1 milligram per kilo in the left, 2 milligrams per kilo in the center, and 4 milligrams per kilo in the right. And the reduction rates of heparan sulfate concentrations were lower in the 1 milligram per kilo group than in the 2 and 4 milligram per kilo group. And all patients in the 2 and 4 milligram per kilo group, they have decreased the levels of heparan sulfate in the CSF, showing that the enzyme really crosses the blood-brain barrier. Let's see the somatic effects. Dermatan sulfate in serum. You see they are basically, these are the upper part of this slide is the patients that were switched from idursulfase. You see they basically maintained the levels of dermatan sulfate in serum. And the patients who were naive, there was a sharp decrease on the levels of dermatan sulfate in the 2 and 4 milligrams per kilo group. Regarding liver volumes, the same. You can see basically the patients who switched from idursulfase, they maintained the liver volumes along the study, and the patients who were who were naive of therapy, the 2 and 4 milligrams per kilo, they show a decrease in liver volume. So the summary of this study, JR141 was tolerated at the maximum dose of 4 milligrams per kilo. The levels of heparan sulfate in the CSF decreasing in all patients in the 2 and 4 milligrams per kilo group. Developmental age was stabilized or improved in most patients. I did not show these results. Efficacy and safety on systemic symptoms were comparable to current enzyme replacement therapy in this 2.0 milligrams per kilo group. So the conclusion, the weekly administration of JR141 at a dose of 2 milligrams per kilo was considered optimal for patients with MPS II. Now I will show some results that combine data from Brazil and Japan, like this. In severe patients, severe MPS II patients, the CSF levels of heparan sulfate, they decreased in these patients, and you see that they decreased below 4,000. I put in red the 4,000 because this is the upper level that you find in patients with attenuated MPS II. So if a patient can have up to 4,000 in terms of heparan sulfate and having attenuated MPS II, this is a kind of safe limit. And so all patients, the heparan sulfate levels in the CSF decreased in all subjects on intravenous enzyme replacement therapy with JR141, confirming that it passes the blood-brain barrier and has long-term substrate degrading effect in CNS. You see that there are most patients that have follow-up of over 2 years on this. And this was already shown by Professor Eto, but I want to highlight that in this first part in the left is the patient on the phase 1 that received the enzyme replacement therapy with this fusion protein, and then there is a decrease of CSF heparan sulfate after just 3 weeks, and then they stop the therapy. They come back to the standard intravenous therapy with idursulfase, and the levels of heparan sulfate in the CSF went up again, as you can see in the middle part of this slide. And then they start the phase 2-3 study, and then there was a sharp decline on the levels of heparan sulfate and a sharp and sustained decline along the duration of this study. So this is quite nice to demonstrate that the enzyme, the BBB penetrating enzyme, has an effect on the CSF. And regarding the developmental assessment, this was already also shown by Professor Eto, but I want to highlight that most patients, they stabilized, the developmental assessment was stabilized or improved, suggesting that JR141 may improve CNS symptoms in MPS I. You see the trajectories, the dots connected by a line are the same patients. You see that most patients, especially the ones treated earlier, they have a stabilization or improvement in the developmental assessment. And in this figure, you see the changes seen over 52 weeks in 3 subdomains of the Vineland. And you see that in green is the patients that improved, in blue the patients that stabilized, and in orange, the patients deteriorate. And then see that most patients, they or improved or stabilized along the study. So these are very encouraging results. This was already shown by Professor Eto. I just emphasize because I think this is very interesting. This was already presented in a symposium, but it was already accepted for publication. And you see that in blue, you see the patient that was started therapy by about 2 years of age with standard intravenous enzyme replacement therapy with dorsal phase, and you see that the patient does not follow the normal development, but the sibling that was treated around 1 year of age with the BBB penetrating enzyme, he's following exactly the normal development. So the importance of treating early and treating with the enzyme that reaches the CNS. So these studies on the preliminary studies on the GR141, they already published the phase 2 in Brazil. And I want to stress that here you see my colleague Ana Martins in the site of São Paulo and our site in Porto Alegre, and also the study of the phase 2/3 trial in Japan was already published, and Professor Okuyama, Professor Eto, Professor Sakai, and Professor Nakamura have sites on this study. So let's now move back to MPS I, because JCR has a program on this MPS as well, and the name is JR171. It's the same approach. You see the standard enzyme replacement therapy does not bypass the blood-brain barrier, but the one with this fusion protein is able to cross the blood-brain barrier and reach the brain. And I want to show you these preclinical results. You see here in the left part of this figure, you see that in black, the untreated mice, and in purple, you see the mice treated with laronidase. You see that the levels of GAGs in the CSF in the brain, they are increased. But when they are treated with the JR171, you see in the 3 columns in the middle, they are very low, very close to normal. The same you see in the brain. And but when you go to somatic part, you see that JR171 is comparable to the laronidase. So showing that the difference is that the one enzyme, the fusion protein, can reach the brain and can decrease the levels of GAGs there. So and this is the slide of the part 1 of the JR171 clinical trial. You see patients. After 2 weeks, all the 4 patients treated, they decreased substantially the levels of GAGs in the heparan sulfate and the central nervous system and the CSF. So this was very encouraging results. Now the study is moving on, and I hope in the next meeting you will have data to present about this study. So summarizing what was developed in this area, first we had the company Armagen using the insulin receptor, working in MPS I. They completed a phase 1 trial in Brazil, but the Armagen is now part of JCR. Then you have this Japanese company JCR. They're working with the transferrin receptor, first addressing MPS II. They completed a phase 1/2 study in Japan. They completed a phase 2 in Brazil, also completed a phase 2/3 in Japan. And in fact, this product is approved in Japan, becoming the first intravenous BBB penetrating enzyme approved for MPS. They are now starting a phase 3 trial, which will be a global trial involving, in addition to Japan, Brazil, also North America and Europe. Also, they are now starting a trial on MPS I It's a phase 1-2 trial to be performed also in Japan, Brazil, and other countries. And they're in preclinical. They have studies on MPS IIIA, MPS IIIB, and MPS 7. Tomorrow at the poster session, which starts at 4 p.m. Central European time, there are 2 posters about these studies, the MPS 1 and MPS 2, poster 47 for MPS 2 and 48 for MPS 1, so you'll have more data on these studies. And I also want to highlight there is another company, a North American, US company, Denali. Denali also uses the transferrin receptor and is working with MPS 2. It's in early clinical development, and they have plans also to work on MPS IIIA and MPS IIIB. So it is an evolving field, and I'm very pleased that we have nice results to show about this technology. And so I wish to thank you very much. And here is my email address if you have questions, but I'm ready to answer questions now. And I wish that in the future we'll be able to have another face-to-face meeting, maybe in the next MPS meeting. I have very good memories of the one we organized in Brazil in 2014, and I hope that we can have again this very important meeting with professionals and families about the new discoveries on the MPS film. So thank you very much. I'm ready for questions.
Thank you very much, Professor Giuliani. It was extraordinary work in these new technologies, new therapeutics. There is a question in the audience that says, what were the inclusion and exclusion criteria of the patients? This question arrived at 10 minutes of your presentation, so I think it was directed to the first part of the MPS 1.
Well, this trial was, thank you for the question, it's very inclusive. They included patients with severe disease and also with attenuated disease, patients that were on previous enzyme replacement therapy, provided they were in a stable dose for a few months at least. Or they could be naive. What was in exclusion is to participate in another clinical trial, like gene therapy, for instance, or having previous hematopoietic stem cell transplantation. I think these are the most important. Or also to being unable to have a lumbar puncture also is an exclusion criteria, because you need to measure the levels of GAGs in the CSF. These are the main inclusion exclusion criteria.
Okay, there is another question also that is, can you give more details on the studies on MPS 7 ERT to cross BBB?
Well, I think that this trial, this is still on preclinical development. It is on this website of JCR as in the pipeline, but I think it's in preclinical development at this moment. I do not know about the plans for clinical development. I'm not aware.
Okay, there is also a question coming from Bangalore, India, asking if a patient with MPS 2 could have the opportunity to participate in a clinical trial of GCR.
Yes, there is a new trial on a phase 3 trial for MPS 2 that will start shortly. And patients with MPS 2, they will be able to participate depending on reaching the site that will enroll patients.
Okay, and also in addition, what are your thoughts of the GCR product for MPS 1 compared to the hematopoietic stem cell transplantation?
It's a very, very good question. I do not have an answer because the trial is just starting. I think MPS, the transplantation, you provide a successful transplantation, you provide an amount of enzyme to the patients based on the cells that are transplanted, but it is a small amount, but it's a continuous enzyme. In the intravenous enzyme replacement therapy with the brain penetrating enzyme, you provide a pulse of enzyme every 7 days. And so we have to follow to see the advantages and limitations of each therapy. But maybe I think this is something that you can think on combine both therapies. I think that as there is a proposal to combine hematopoietic stem cell transplantation with intrathecal ERT. Maybe they can combine with intravenous ERT with a brain penetrating enzyme, at least for some time, while until the transplantation is fully operating. But also we have to consider the risks of transplantation, which are not that high in North America and Europe, but in other regions, these risks are quite important. And in Brazil, we have a mortality rate that is around 30%, which is quite important. So always a concern, this hematopoietic stem cell transplantation in terms of safety. But in terms of efficacy, we will have to show in the long term how they perform.
Thank you very much, Professor Giuliani. But now we still have some questions that I will direct to you. But now we will pass to the next speaker, which is the Professor Paul Harmatz. So, Professor Paul Harmatz, hello. How are you today? A little bit tired because of the time, I guess.
Yes.
So Professor Harmatz is the professor in the Department of Pediatrics in the University of California, San Francisco, California, USA, and is a professor in residence, Department of Pediatrics, University of California, San Francisco, and UCSF Benioff Children's Hospital in Oakland. He is the medical director of the Pediatric Clinical Research Program in mucopolysaccharidosis and related disorders. During the last 20 years, Dr. Harmatz has participated in clinical trials developing therapies for many of the mucopolysaccharidosis, Mps 1, 2, 3A, 3B, 4A, 6, 7, and has managed clinical care for patients with MPS living in the Northern California. So today, the presentation of Professor Harmatz is the update on enzymatic therapies. So, Professor Harmatz, the floor is yours.
Thank you, Dr. Pinto. It is an honor to present. I thank the organizers. I recognize that, just as Dr. Giuliani mentioned, that we look forward to traveling. Hopefully in 2 to 3 years, we will be through this pandemic time. And it will get me into a normal time zone, and it's much more enjoyable to meet families and other scientists and physicians than the virtual platform. But it's a wonderful program. It's really filled with a lot of new material and exciting progress. And it sort of shows maybe every 3 years is a better time frame than every other year, because the progress has been remarkable since the last meeting. My disclosures, I do recognize when I'm a pediatric gastroenterologist, so I'm not a geneticist. I didn't grow up in this field. I was rescued from BioMarin from pediatric GI, and the diseases diarrhea, constipation, liver disease, very similar to Fritz Weiberg. He came from the same background, but it's been a really rewarding, exciting field. But in the past, when we first presented, I would always be on the program number 3 after Lauren Clark presenting MPS I, Joe Munzer presenting MPS II, and I was then following with MPS VI. So this was all of the background and much of the experimental processes were completely summarized by Dr. Clark and Munzer before I rose. And today is very similar. Following Dr. Ito and Dr. Giuliani is a really hard task. And I find little niches. I'll repeat some of the material that they said, but tried to stay away from as much of the fusion protein, which is very exciting at this time. So today I'll summarize a bit about how much progress, which is really remarkable. I think we are excited about the future, but we have to recognize what has happened in the last 20 to 30 years as truly remarkable, the number of new therapies that have emerged. And I'll mention a bit about enzyme therapies that are ongoing, that we have high hopes that this will contribute and help stabilize patients and give them an opportunity to wait for really definitive cures and therapies, particularly in the gene therapy areas. And I'll finish with just a brief run through of lessons we learned about clinical trials from MPS VI, and hopefully these lessons are helpful to the new trials that, especially that Dr. Giuliani has been summarizing, and are really unique to trials in ultra rare disease, where you have to deal with extremely heterogeneous small populations and still follow at least in the US, the federal regulatory option or requirement that we produce a well-controlled clinical trial for approval. So this really summarizes when I came in the field in 2000 and where we are in 2021. And you can see in 2000, we had supportive care. We tried our best to provide surgeries that would stabilize the spine, decompress the spine. You heard about many of these surgical procedures and respiratory support procedures yesterday in the symposium. We treated acute infections. We tried to control pain. But stem cell transplant was the one specific therapy that we had at that time. And then in 2021, we now have, as Dr. Pintos summarized, we have 5 FDA-approved therapies and a lot of hope and improvement in the clinical condition in the patients with these diseases. Coming back, following Dr. Ito's, I've been in the field for 20 years. I'm probably at the end of my academic or working career, but I always look back and try to recognize the people that really set the stage and moved us into this field and made it possible to be where we are today. And the person to recognize, and the 3 or 4 people that I recognize are not the full complement. I'm sure I know I'm leaving many really remarkable scientists that helped move us to the present. But Elizabeth Neufeld was really the critical, I call her the grand dame, the MPS field. She described the cross-correction, which is the breakthrough that really underlies enzyme replacement therapy. Stem cell transplant is probably dependent heavily on cross-correction and not just local production within the cells that have migrated, but and then gene therapy, many of the gene therapies really depend on production, high levels of enzyme and cross-correction. So this was the breakthrough. And they always tell the story that this was a serendipitous accidental mixing of Hurler and Hunter fibroblasts and a post-hoc noticed that the cells had cleared, and was Dr. Neufeld and the postdoc were sharp enough not to disregard it as an accident, and this was a breakthrough discovery. We have activities in between that included, as Dr. Ito said, the development of enzyme therapy for Gaucher as the first enzyme therapy. We have transplant emerging in the 80s as well. And then Emil Kakas really was a breakthrough person for the MPS field. And he, recognizing he did his PhD with Elizabeth Neufeld at UCLA at that time, and came back to Harbor UCLA, finished his residency in medical genetics, and his young faculty project was he had worked heavily with a dog model for MPS I and enzyme replacement therapy. He then carried that into the human trials, and as Dr. Ito showed, he worked in a Quonset that set up as a GMP laboratory, stayed 1 to 2 weeks ahead of where he was in the clinical trial, and was desperate to move forward with a industry sponsor who had the resources to really expand trials, commercialize the drug, and that's BioMarin came into the picture. So this was the breakthrough person. He still leads much of the sort of ethos and conscience and momentum of all of the rare diseases, and including MPS at his company, Ultragenyx. Looking at this group of therapies that we mentioned, it was a very unique merger of really unique basic scientists, and Liz Neufeld holds the top of the list, but Bill Sly, John Hopwood, Emil Kakas, Joe Munzer all drove forward enzyme therapies from a basic science standpoint, laboratory, their own laboratories, developing animal models and pushing in sponsors to move ahead with these therapies. So I'm standing on their shoulders, and any one of them could give this talk much better than me. Where are we? You've heard many exciting new directions that Dr. Ito mentioned and that Dr. Giuliani mentioned that that are already in the trial, and Dr. Avasolo, who's also working at the forefront and presenting a new way that enzyme therapy will move forward. But we all recognize we don't have therapies for all of these disorders, and it's very discouraging for the families that have not come to trial or commercialization, so everybody can benefit from the therapy. And we know that it is tedious and it's expensive to have weekly infusions. One factor that hasn't been mentioned is the effect of the immune response on the enzyme therapy, and this will be an issue that carries forward, but with gene therapy possibly, but most surely with the blood-brain barrier penetrating enzymes. And we don't have a trial of on the immune response to try to modulate it, but this hopefully will be on the sort of forefront of what we're doing in the near future. The skeleton, as Dr. Tomatsu discussed yesterday, the skeleton is poorly responsive to enzyme therapy, and we recognize we need more newborn screening programs. It's incredibly slow in the United States, Europe as well. It's rewarding to see it move forward so quickly in Japan. So this is hopefully will accelerate in the next 5 years. And the CNS has been the hardest organ to treat, and now we have all the focus is moving towards how to treat the brain. Most of my time just on some small issues that might help with IV therapy, and then summarize two trials that are similar to what Dr. Ito described in Japan with intrathecal and intraventricular DRT. One question that comes up whenever we're giving enzyme infusions, and generally we tried to stay with about the time frame that was used in the clinical trials is the best evidence we have of efficacy, but it's been, people are always asking, can we shorten the infusion time? And the patients are anxious to move out of the infusion center. It's a little bit easier with home infusions, but there was one publication combined BioMarin and Mark Haskin's lab looking at the CAP model, comparing 2-hour infusion versus 4-hour infusion. And they do show a pharmacokinetic difference. You do get more area under the curve with a 4-hour infusion. Same dose was given at both time points. But when they looked in detail at the cats after 6 months, they could not see a difference in the bone effects, the urine gag, and storage of gag in various organs. They did not look at the brain, but it looked, at least in the CAP model, that 2 hours was equally effective and provides some basis if people are shortening their infusion times. Dr. Giuliani and Dr. Horowitz both had papers looking at MPS I patients. Dr. Giuliani compared standard dose to higher dose, saw similar efficacy, but more side effects, infusion-related reactions in the high dose. So we've generally stayed with the standard dose unless we're seeing poor efficacy and are really desperate to try higher doses. Dr. Horowitz compared weekly with every other week. Weekly was used for a year, then the patient shifted to every other week at double dose, and she really saw a very similar clinical picture and supported the possibility that every other week could be considered. And then finishing with the slide, I mentioned again that immune modulation really has to be at the forefront of our thinking at this point. Dr. Priya Krishnani has really laid the groundwork. There's been a few other papers looking at different immune modulating protocols, including Dr. Giuliani's, but Dr. Krishnani, working with Pompeii, has been able to assemble a pretty standard and effective way to immune tolerize when you administer the medications at the same time as the antigen, as the enzyme is introduced. And but it's also possible to modulate someone who's not responding to enzyme therapy. And again, you have to look at biomarker. We don't have a good feel, except for Dr. Tomatsu, of circulating gag. We're usually following urine gag. But if we see a patient with poor clinical response, very severe mutation, and a urine gag that either did not drop or dropped and then returned to starting levels, and particularly if they have high antibody, high cell blocking antibody, binding antibody, it's worth considering this. And there are 2 papers in the literature to give you a sort of base to move off of in MPS, in one case with an immune modulation with Dr. Burton's paper, and then Dr. Krishnani and the group in Phoenix had a 3-year-old who had not been exposed to enzyme, had a severe inversion mutation, and was immune tolerized and had a very good response of urine gag and good clinical response. So I think this is probably the biggest take home, is that the antibody could be having a significant effect in a small number of very severe patients and needs to be considered. Another breakthrough in the last 5 years was the approval of enzyme for MPS 7. And it just gives you an example of how Emil Kakkis was able to move forward with a disease that is incredibly rare compared to MPS II or even MPS VI, although Dr. Ito points out he has very few MPS VI in Japan, but there probably are in the 100 to 200 patients in the world, and 10 were in the US at the time we tried to move to a phase 3 trial, and it took an international effort to recruit for a 12-patient trial. And what you're seeing are a unique design that included a blind start, randomized trial where we started 3 patients in each group, giving us the 12 patients. It took away bias, it added power. It was not a classic randomized double-blind placebo control, but close, and was able to support approval. The other unique feature of this trial was the use of multiple endpoints and developing a multiple domain response index that is shown in this diagram. These patients are so heterogeneous that some could walk, some could not walk, some could do the pulmonary function, some couldn't. And you would basically rate each patient individually and look for overall improvement, that it may be in a completely different domain for each patient. So, intraventricular, we're in the midst of 2 trials in the US. You're well aware of the Shire and Takeda trial. It's led by Joe Munzer. This data was presented at the World Meeting. There has been over a several year period a phase 2-3 study, and sub-studies with siblings being compared and patients less than 3 years old being studied. And this was a trial set up with patients who were 3 to 18. They had a fairly tight DAS score in the 55 to 85 range, but it was still a when you take an age range of that, you have slowly progressing, you have rapidly progressing patients. A very difficult trial, and you have 40 patients, 2-to-1 randomization, followed for a 3-year period now. And a similar setup for the under 3. There are a couple of different groups based on age and dose, but they were basically all started on drug and then moved to an extension trial, and there are 9 patients in this group. Showing you the data for the phase 2-3, it's still in progress. Patients are still all receiving drug. The data is being analyzed, and you can see when they look at select age groups here, particularly the under 6 age group appears to have a positive response, and you're seeing the treated group in red, the placebo or observational. They were not placebo with observation for a year, so they had a delayed start, and they have a more rapid drop in their developmental quotient, the GCA score, and the DAS. So you see rapid, then a stabilization, similar drop. It doesn't stop the change in the GCA score over time, but these patients, it's important to point out that this may be a maintenance of developmental level, and the drop is related to the change in age in these patients. So the safety was, the drug was well tolerated. The device was probably causing more issues in the study than the drug. And I'm not showing the individual data from the under 3 study, but the 9 patients all stabilized. They used Bailey in the early age range, then they moved to the DAS2 when they reached a certain age, and there is a change in score level, but the patient stabilized. So the conclusion is that we can get an improved outcome in select older patients and probably most younger patients that who are started before severe injury has occurred. A second presentation credited to Nicole Musval and provided by Elevex is the examination of the enzyme for Sanfilippo B. This enzyme is a IGF-2 fusion protein. It's significant dose of drug. You can see that within short time frame, this is unaffected CSF level for heparan sulfate NREs, non-reducing assay. This is untreated and then treated with drugs. So very rapid drop to normal levels, improvement in liver size documented. You can see the change in brain volume pre-treatment and during the first 6 months, and then later in the therapy, the brain volume is actually beginning to improve. So probably loss of brain volume, loss of storage material, and then improvement in brain volume. And when you plot the change in brain volume, the change in age-adjusted scores, you can see improvement in outcome, very promising. The overall, yes, the improvement in the natural history shows that we do get in a significant percentage stabilization improvement in the rapid progressors, and in the slow progressors we stabilize their cognitive scores. So their age equivalent scores, they're stable, they're maintaining their functional level. So I summarize, this is the treatment outcomes. So as I said, the heparan sulfate drops to normal, the liver reduces size, the brain is showing increasing size after the first 6 months, and cognitive scores look to be improving. I'm going to not go in depth. I would just emphasize that a large animal model was critical. A survey study that was unique, that defined a biomarker that I have great trust in, which is the urine GAG in MPS VI, really defined 20 years as the lifespan for a rapidly progressing patient. You really want to, the better biomarker you have, the better we're going to be able to study these diseases. Mutation analysis was done on every patient in those 100 survey patients. This was at 10% of the population. We had good outcomes on a 12-minute walk, picked the two difficult primary outcomes. It's hard to maintain after the trial. But the thing to be aware of is that we had a unique statistician who was able to correct for a walk difference at baseline of over 100 meters. So you would really like this to be identical and find a way to stratify your populations so you get equal patients in both groups. And then long-term follow-up, I really suggest you have a good survey early, and then 10 years later, 5 years later, follow those patients with the same rigor, and it can lead to data that suggests that you have increased survival. So this is the treated MPS VI patients. These are the untreated patients, and this is really the most important outcome. Dr. Ito mentioned sibling studies. Every sibling study is critical, helps us establish that this early therapy is the best. And this is the Australian Guild. Japan has a wonderful sibling study also. Screening for all of these patients. And Michael Gelb is our pioneer at developing assays. But just to show you the person that's really aged in this picture is me over 20 years with one of our leading patients that and Isabella is 7 here. She's testifying to Congress concerning a immigration issue that had arisen. So she's in Washington providing her testimony to the congressional representatives. She's 6 now. She's survived much longer than we would have expected, in good health than before enzyme therapy. Just finally closing to thank the patients. They did and they continue to do an enormous effort to help us through clinical trials. We would not have therapies without the patients willing to travel or relocate. And thank John Hopwood, who's hopefully listening into the meeting. And in Australia, he was the pioneer in this disease, and the patients loved having him visit and talk about their how he brought this through the development stage to a product. So thank you.
Thank you very much, Professor Harmatz, for showing us so nicely the evolution of the different treatment for mucopolysaccharidosis that I think is very, very important area in the field. Do you think that with all these different types of treatment, we perhaps could achieve this kind of personalized medicine where different types of treatment may be applied for different populations of the MPS?
I think you're correct, Dr. Pinto. Dr. Giuliani, Roberto mentioned one aspect of that, which is combinations. It may be a combination in which 2 different ways of presenting enzyme. It also may be a combination in the future where we do have trials of inflammatory agents and substrate reducing agents that are ongoing. And it may be that these molecules with different size and function will complement each other. But the third aspect, I think that we're probably overlooking the effect of antibody on the effect, how well the therapy works. And we may find that we have to use it to improve transfer of IV enzyme into the brain. So I think that, yes, it'll be very personalized. It'll be hard to do trials in those areas to prove that the combination is better, but I think the physicians will work that out with each patient.
Okay, so I think we reached the end of our time, and I would like to say thank you to all of you, to all the presenters of this round table, and also thank you to the MPS organization for this very nice group of excellent presenters. Thank you. Thank you very much, and see you soon. Bye.
Thank you, everybody.
Bye-bye.